What do you get when you cross a vampire and a teacher? A vampire and a teacher. Blood tests. No, you don't like that one? What did one eye say to the other? What did one eye say to the other? That's like something like this, right? You know this one? It'll come to you. Sniff it out. Just between you and me, something smells. Okay. What is a frog's favorite kind of flower? What is a frog's favorite kind of flower? A crocus. Nicely done. You should be proud of yourself. A crocus. Okay. Some housekeeping stuff. Quiz 12 is due this Friday by midnight. Quiz 12, due Friday by midnight. Review sessions. We'll put these on Zoom or on Canvas, but Friday 2 to 3 by Zoom. Friday 2 to 3 by Zoom. Monday 2 to 3 by Zoom. Monday 6 to 7 by Zoom and in Bio 260. Monday 7 to 8, Science Annex 117. Tuesday 11 to noon, Science Annex 117. Okay? So you've got 5 review sessions for exam 4, which is next Wednesday. Got it? Monday 2 to 3, 6 to 7, 7 to 8. Yep. Science Annex 117. Okay. Yes.
Will those review session times be posted?
Yes, we'll put those on Canvas as well. They're probably going on Canvas right now, I'm guessing. All right. Your exam or your semester grade is current and accurate. There is a what if option in Canvas where you can kind of input your scores, like a what if tool or a what if score. I'd encourage you to look at that. It's not going to drop your lowest. So you might have to do a little mathematics, or you can come see me next week in office hours. I saw a few of you today and yesterday. We'll have a bonus opportunity at the end of the lecture today, so I'm glad you're here. After you start that tonight, your grades will get a little wonky again, okay? Because the bonus opportunity on Canvas counts it out of the points that I assign. It doesn't make it out of 0. So we have to do some math behind the scenes and get rid of those bonus assignments. That's why they disappear, and add them to your exam score. I can't do that until you take the exam. Okay, so after exam 4, I'll have another announcement saying, hey, Canvas has been cleaned up, the grade book should be accurate. Then you can start making decisions about, am I going to take this final or not? If you're not going to take the final, remember, you still have a participation opportunity in class, and you still have a participation opportunity for a review session. Those are worth 10 more points. So if you're going to not take exam 5, I would still encourage you to come to class, get the participation points, go to a review session. Most importantly, because you actually need to know this material. It's not just about getting out of an exam, right? Yes? There'll be a bonus opportunity for exam 5, but it won't count unless you're taking exam 5. Yeah, make sense? So it'll kind of be part of exam 5. For those of you that have had 4 solid exams, you're going to drop your lowest, which is exam 5. You're not going to get the bonus opportunity on exam 5. Those rules make sense? Guidelines make sense? Okay. All right. This is where we need to pick up. How many of you did the homework that I asked you to do, which was look ahead a few slides. Okay, good. Good. So we're going to walk through the rest of the NMJ, the neuromuscular junction, and then we're going to move directly into the sliding filament model. You will see that there's only 2 lectures posted on slides. So we'll start lecture 2 today after we get through lecture 1, and we'll get most of the way through it. And then on Monday, we'll finish up lecture 2, and that content is on the exam. But the more challenging content was Monday and will be today. Next Monday, kind of the complexity will go down a little bit. The sliding filament, muscle contraction, we'll watch a video today. That is the most challenging concept in this particular unit. So if we look at the neuromuscular junction, we can see that there are opportunities for certain pharmaceuticals, certain drugs, certain environmental toxins like pesticides to actually bind and inhibit at the neuromuscular junction. So they can paralyze the muscles, and pesticides that contain cholinesterase inhibitors, they bind to acetylcholine esterase, and they prevent it from degrading acetylcholine. If you don't degrade the acetylcholine, what happens to the muscle? If you never remove acetylcholine, what happens to the muscle? It stays relaxed or contracted? Contracted. So we call that spastic paralysis. It's paralyzed in a contracted or a spastic form. So in those situations, the unfortunate clinical result for that organism might be suffocation. And that's how pesticides work. If the insect is using acetylcholine, it paralyzes in a contracted state, so you can't relax the muscles. Another example of spastic paralysis, Clostridium tetani. This is a tetanus. You get a tetanus shot if you step on a rusty nail. Lockjaw. So the muscles within the jaw are particularly sensitive to Clostridium tetani, and it freezes or paralyzes them in a contracted form, and you can't open up your jaw, right? Glycine in the spinal cord normally inhibits motor neurons from producing unwanted motions. That's why when you move, it's not like jerky. It's nice and smooth because of glycine's activity. So this tetanus toxin blocks the glycine release in the spinal cord, and it causes this overstimulation and spastic paralysis. So 2 examples on this slide of spastic paralysis. That's a pretty important term to know. I've given you 2 examples that I would know for next Wednesday. In contrast, let's look at flaccid paralysis. Without reading it, who can tell me what that is? What's flaccid paralysis? Real quick. The muscle is stuck in a relaxed state. Flaccid, that's what that word means. Flaccid paralysis. So curare is a very common substance harvested out of certain plants and certain organisms, like certain frogs in South America, and is used to coat the tip of an arrow. And these indigenous groups of people use this for hunting, or they use it for defense, right, against other tribes. So they'll coat their tips, their poison blow darts, and they'll literally blow dart into an animal or a foe, someone from the other tribe, and it causes flaccid paralysis, and the organism can't run away. So curare competes with acetylcholine receptor sites, but it doesn't stimulate the muscle. So it's competitive inhibition, is that terminology. It binds to the acetylcholine receptor sites on the other side of the cleft, It blocks the ability for acetylcholine to bind, but it doesn't stimulate muscle contraction. It doesn't propagate an action potential on the motor end plate or the other side. Another example that we'll bring back, we talked about this earlier in the semester. Do you remember the Botox conversation? So botulism is a type of food poisoning. This is caused by a neuromuscular toxin, also an example of flaccid paralysis. And the toxin that's made by the bacterium is Clostridium botulinum. This blocks acetylcholine release, so you don't have any neurotransmitter crossing the cleft. The muscle's stuck in a relaxed phase. Now, some entities have gotten clever, right? Some industry applications. And Botox is being used for relaxing facial muscles and therefore eliminating the appearance of wrinkles. There's another more recent indication for use for Botox. Anybody know it? How is it being used clinically? It's not for vanity or cosmetic applications. Migraines, that's right. It's being used with a tremendous amount of success to relax the muscles of the face and the cranial cap to alleviate that pressure for people that suffer from aggressive migraines. And they'll get numerous Botox injections, and it's on label. It has an indication statement for that. It's actually quite successful. Questions? Kind of finishes out neuromuscular junction. Okay, now we get into transition. We get into really the core of the muscle physiology. And we're going to talk about the sliding filament model. And these are the slides I really wanted you to take a look at. So I'd like you to appreciate the features of this electron micrograph. They've been labeled for you. You can see the Z lines, very prominent. And Z line to Z line is described as what? Z line to Z line or Z disc to Z disc. What is that? Just shout it out. Sarcomere. Thank you. What's the definition of a sarcomere? Not in the original Greek, but what's the definition of a sarcomere? But you can tell me the original Greek too if you're curious, if you have that. Pardon? It is the portion between Z line and Z line. That's correct. There's a definition of functional unit of muscle, the sarcomere. Remember, it translates to what? A flesh part, right? Do you see these Z lines here in a relaxed state? Do you see the Z lines here in a contracted state? The Z lines move closer together, and therefore the thick and the thin filaments overlap. And we're going to unpack that a little bit more, like in this slide. So you can appreciate a relaxed muscle. Here is a similar type of electron micrograph on your right. Here is a schematic or a cartoon drawing of what's happening on the transmission electron micrograph. And you can appreciate the M line. You can see the thick filament in purple, the thin filament more in this orange color. And as the muscle contracts and the Z lines approximate each other, that's just a word that means they get closer together, until it's fully contracted and the Z lines are at their closest proximity. They're the closest they're going to get. But yet, they bottom out. There's maximum overlap on the thick and the thin filaments, and the thick filament can't contract any further. Make sense? So mechanistically, this is a relaxed state up top. This is a contracted state down low. A contracted sarcomere. And that, folks, is really the secret to muscle contraction. Now, what makes this happen? This is the relaxed starting point. This is the contracted result. Now we're going to walk through an overview, and then we'll come back to it in more detail and talk about these steps. So this is an overview, but I wanted you to get the overview, and this is what I wanted you to walk through initially before today. So if you didn't do this, we're going to do it right now. We're up top. This is what? What is this structure up here? What's coming down? Is it an efferent or an afferent neuron? It's an efferent neuron. So it's a motor efferent neuron. Is it myelinated or unmyelinated in this picture? It's myelinated. It innervates onto a muscle. And at the point that the nerve contacts the muscle, we call that what? That junction. What's it called? Neuromuscular junction. We have a depolarization event that comes down. And you can explain to me that depolarization event, what happens with sodium, what happens with potassium. Right? That was the last unit. So a depolarization event opens up a sodium gate. Sodium comes in. You depolarize. You make the inside of the nerve less negative, more positive. Ultimately, you cycle potassium as well. But what you do at the synaptic knob is you open up a voltage-sensitive calcium gate. The action potential is the voltage. Instead of opening just a sodium voltage gate, it opens a calcium voltage gate. That calcium voltage gate allows calcium to come into the nerve from outside of the cell. That stimulates the release of these vesicles storing acetylcholine. They dump the contents into the cleft, and two acetylcholine molecules in green bind to acetylcholine receptor on the other side. When that happens, that acetylcholine receptor opens up. First, it allows permeability for sodium. Sodium comes into the cell. Where are we? What membrane is this? Is it the nerve or the muscle? It's the muscle. See the word sarcolemma? That's the phospholipid bilayer or the plasma membrane that's specific to the muscle. So now sodium enters through this acetylcholine receptor, causing a depolarization event on the muscle. That opens voltage-sensitive sodium gates, followed by voltage-sensitive potassium gates, just like we studied with the nerve. Make sense? We'll do this again, don't worry, and I'll have bigger pictures. We'll walk through it again in more detail. Now, we're at the level of the muscle, and we have an action potential that's traveling down the muscle. The action potential came from the nerve, and then it had to cross the chasm using acetylcholine. We're on the other side. We call the other side the motor end plate. It's an electrical term. That's why it's referred to as the motor end plate. It's basically just the muscle. The motor end plate is the muscle side of the junction. The motor end plate is the sarcolemma. It's the plasma membrane on the muscle side, where you're opening sodium-potassium gates to propagate that action potential because it went from nerve across the synapse to the muscle or the motor end plate. We call it the motor end plate partly because it's a motor efferent pathway that we use. That's where we get this terminology. So now this action potential can come down the sarcolemma, dive into the T-tubule that we learned about on Monday. If it dives into the T-tubule as an extension of the plasma membrane or the sarcolemma, you're getting the action potential deep into the cell. It's not just at the surface. It actually comes into the cell. Because into the cell, in the belly of the cell, is where the myosin and the actin sit, right? As it comes down here, it triggers the opening of calcium gates in the sarcoplasmic reticulum on either side. Do you see why the triad is so important? You have an action potential that comes down from the surface of the sarcolemma, dives into the T-tubule, conveniently on either side of the T-tubule are the terminal cisternae of the sarcoplasmic reticulum. The terminal cisternae, we said on Monday, translates to end tank or end reservoir, like a cistern is a reservoir. Usually a cistern is a reservoir of water. Here it's a reservoir of calcium. That calcium leaves out of the terminal cisternae, which is just part of the sarcoplasmic reticulum, and it floods into the cytoplasm of that particular cell. And that's the muscle. In the cytoplasm of that cell, it bathes the thin filament. It finds troponin. It binds to troponin, causes troponin to make a conformational change. Tropomyosin that's physically attached to troponin is pulled out of the way, exposing these binding sites for the myosin head. This myosin head is going to find the binding site on the actin molecule. Make sense? Story doesn't end here, but what we've talked about so far is action potential down the nerve, crossing the synapse, triggers an action potential on the muscle, also known as the sarcolemma, the plasma membrane, the motor end plate. That action potential travels along the muscle sarcolemma, the muscle membrane, finds a hole, turns, makes a right turn, a right angle, dives deep down the T-tubule into the cell, the muscle cell. On either side of the T-tubule are the sarcoplasmic reticulum tanks, known as the terminal cisternae, that house the calcium. That electrical signal down the T-tubule opens the calcium gates. Calcium floods into the cell, finds troponin. All of this is happening just so you can scratch your cheek, right? Every time a muscle contracts, this happens. Isn't that crazy to think? I haven't even finished what happens every time the muscle contracts. And you don't even think about this. You're not like, okay, action potential, all right, sodium, all right, potassium, acetylcholine, esterase, get it out of there, calcium on the other side, right? Troponin, get out of the way, tropomyosin, right? Here we come, myosin head, woohoo, power stroke, relax, scratch again, right? So we're here. Now we go through this cycle of how the actual sarcomere contracts. It's a cyclical pattern. I'm going to show you a video so you can kind of appreciate it in more of an animated phase. But we're going to start right here. When it binds, we call the binding of the myosin head, which is on the thick filament, to the actin molecule of the thin filament, we call that binding cross-bridge formation. That's the terminology. So now we have a cross-bridge that formed. The cross-bridge forms allows this to interact. Then down low at the 6 o'clock position, you can see what we refer to as the power stroke, is that head fires, stays attached to the thin filament, but the myosin head fires. It fires, which requires energy in the form of ATP, and you cleave off a phosphate group. You have 3 phosphates on an ATP. So here we fire it, and we liberate this ADP, which is 2 phosphates, and the third phosphate that's just kind of hanging around. Now for this to detach after it's fired, you have to bind a new ATP molecule to release it. ATP is necessary for it to let go. Once it lets go, you have to recock the head. To recock the head, you actually cleave one of the phosphates, so ATP goes to ADP plus 1 phosphate. And now it's from this fired position to this cocked position, and it's ready to bind again. So the motion is kind of like this. Does that make sense? It's like a ratchet mechanism of how actin and myosin slide across each other. Or thick and thin filaments slide across it. That's more accurate. Thick and thin filaments sliding across each other. I won't try to trick you on the exam like true or false. It's myosin and actin that are sliding across each other. It's the thick and the thin filaments that are sliding across each other because the thin filament is made up of more than just actin. Make sense? And the thick filament is myosin plus the myosin heads. So these are technicalities, but I want you to understand the difference. This cycle happens where myosin attaches to the myosin binding site on the actin filament. Myosin attaches to the myosin binding site on the actin filament. This is the myosin binding site on the actin filament. All right, now how about relaxation? Like when you're done with the contraction event, what do you think you need to do to kind of get rid of the signal to contract? A couple things, just think through it. You have to turn off the action potential coming down the nerve, yeah. What about all that acetylcholine that's in the cleft? Break it down. Use acetylcholine esterase. And then its compartmental pieces are reabsorbed. And acetylcholine is recycled, essentially. You have an acetyl group and a choline group that's reabsorbed, repackages acetylcholine, put in a vesicle, and it can actually do it again. You don't have to manufacture acetylcholine every time you fire. You just kind of recycle the component parts after the enzyme breaks it down. Okay, so you cease the nerve signal. You resequester or break down acetylcholine, reabsorb calcium back into the sarcoplasmic reticulum. Does that make sense? Reabsorb it so that it's no longer binding to troponin, and you no longer have the binding site available because tropomyosin hides the myosin binding site on the actin filament. Couple of key notes. Acetylcholine destruction or breakdown is from acetylcholine esterase. I told you it makes it into an acetyl and a choline group, reabsorbs it, recycles it, makes a new molecule, puts it in a synaptic vesicle. Calcium uptake. This is another active transport. What does active transport mean to you? Go all the way back to Unit 1. If I said this is active transport, what does that mean to you? It requires energy, also in the form of ATP. You actively pump calcium back into the sarcoplasmic reticulum using this active transport. Calcium has a molecule inside the terminal cisternae of the sarcoplasmic reticulum known as calsequestrin. Kind of a cool name. It sequesters, that's a word that means hold back or reabsorbs calcium. It binds to calsequestrin inside the terminal cisternae and makes the concentration of calcium inside the muscle cell lower because it's storing it inside of an internal compartment we call the sarcoplasmic reticulum. So ATP is absolutely required for two main processes. What are the two main processes? I talked about on the previous slide, and I talked about here. To separate myosin from the myosin binding site on the actin filament, to let go, absolutely requires ATP. And what about calcium reuptake? Absolutely requires ATP. So if you have a case where ATP is no longer being provided, you're going to have the muscle stuck in a contracted state. A rigor state is what we call it. And that's where we get the terminology rigor mortis post-mortem. Okay, after death, individuals that are deceased will become stiff. Their whole body will become stiff because you're no longer making energy. If you're no longer making energy, every single muscle in the body is kind of stuck in a cross-bridge formation with no release. So everything is no longer can slide. And calcium is widely available inside the cell, the muscle cell, because ATP is needed to pump calcium into the terminal cisternae to find calsequestrin. So rigor mortis is a term. It's a hardening of the muscle or a stiffening of the body. Begins about 3 to 4 hours post-mortem or after death. you have a deteriorating or a breaking down sarcoplasmic reticulum, and calcium just floods into the muscle cell because it's starting to decay. That sarcoplasmic reticulum membrane is starting to decay. The sarcolemma also allows, that's the plasma membrane of the muscle, also allows calcium to come into the cell. So you have plenty of calcium around, allowing for a cross bridge formation. So the muscles all can contract, but they can't relax. Muscle relaxation requires ATP, like we talked about, for the cycling, as well as the resequestration of calcium. So that is no longer happening. Rigor peaks at about 12 hours post-mortem, and then starts to diminish over the next 48 to 60 hours. Why does it go up, and then why does it come down? Any ideas? The rigor, the stiffening. Well, the calcium is there, so it's all available, but this thing that's supposed to detach with energy, eventually these proteins just decay, and they just sort of fall apart. Okay, so that's just part of the decay process.
Do you see rigor mortis faster in children or infants than adults?
That's a interesting question. I have never had that question. I don't know the answer to that. Let me think. So you probably would, based upon metabolism, because a smaller or a younger individual, smaller organism is going to have less energy availability. So you would probably anticipate to see rigor happening earlier. All right, this is a corny narrated video, but I like the animation. And I love the music.
We move our bodies by contracting skeletal muscle, while the results of this action are plain to see. The mechanism behind the action is not. Contraction of skeletal muscle tissue occurs when actin and myosin myofilaments slide past one another, causing the sarcomere to shorten. It is important to note that the individual myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs. The molecules you see here are myosin, which at rest has both adenine diphosphate and phosphate molecules attached to the individual myosin heads, actin, tropomyosin, and troponin. When we decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with the troponin molecules. This causes the tropomyosin molecules to move, revealing myosin attachment sites on the actin myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect with the attachment sites on the actin. The resulting formation is called a cross bridge. The remaining adenine diphosphate molecule is then expended in what is called a power stroke, when the myosin heads pull the actin inward. Once the adenine diphosphate attachment is spent, adenine triphosphate molecules attach themselves to the myosin heads. These molecules trigger the release of the myosin heads from the actin attachment sites and are immediately broken down into adenine diphosphate and a single phosphate as the heads move back into their resting position. This is called the recovery stroke. If calcium is still present, the cycle will repeat again, and the sarcomere will continue to shorten until the calcium ions are transported back into the sarcoplasmic reticulum and the muscle relaxes.
Okay, does that help? Just visualize how this works. Fairly complex, but I feel like it totally makes sense. Thoughts? Does it make sense to you how this works? Is it just fascinating that it works this way? Okay, so we're going to walk through this again in the next lecture, even a little bit slower. So you're going to have it kind of twice today. Hopefully 3 times, because I wanted you to review it prior to today. Let's cover a couple of, hang on, let me get my, let's see here. Okay. Couple of practice questions. What surrounds each muscle fascicle or fasciculus, and what surrounds each muscle fiber? What's around the muscle fascicle? Perimysium. And what's around each muscle fiber? Endomysium. Very good. Enlarged areas of the sarcoplasmic reticulum near the T-tubules are called what? Terminal cisternae? What do the terminal cisternae do? Store the calcium. And calcium binds to what molecule in the terminal cisternae? Calsequestrin. Remember that? Okay. Questions? Somewhat clear? Like to see it again?